This is the fourth and final lecture video for this chapter. In this video, we'll be discussing protein synthesis, which is the process in which proteins are made. If you take a look here at this picture, you'll see that the general flow of how genetic information is created, it starts with DNA. Now remember, DNA holds all of the base pairs, and it holds pretty much the blueprint for every single protein that's made in your body. The way that DNA is able to send out these messages through the base pairs is RNA. RNA is kind of like the middleman or the messenger between the DNA and the actual making of the protein. So we're going to talk about this process and how we take the message from DNA, use RNA as a messenger, and finalize our product, which is the protein. So obviously proteins are super important. We've talked about how they are one of the 4 macromolecules. Remember, we have carbohydrates, lipids, proteins, and also nucleic acids. DNA and RNA are nucleic acids, and they are the blueprint for how the proteins are made. Proteins have many different functions. Remember that the most important or the most recognizable type of protein is an enzyme, and enzymes' functions are to lower activation energy. They also speed up the rate of chemical reactions, and not just only enzymes, but there's also plenty other types of protein. And remember that protein make up your hair, your nails, your skin. So there's many different functions, and we've already talked about the importance of eating protein in your diet. But now we're going to talk about how these proteins are made once you've already input them into your diet. So before we talk about the actual process of protein synthesis, I think it's pretty important that we talk about the structure of RNA, the function of RNA, and really like what is RNA? We never even really talked about that yet. We've talked about DNA, which now we all know is a double-stranded molecule in the shape of a double helix. We have nucleotides that are the monomers that make up the large polymer called DNA. We know that it contains a pentose sugar, and the name of the sugar is deoxyribose. We know that there are phosphates. We know that the bases are adenine, thymine, guanine, and cytosine. But what is RNA? I mean, RNA sounds very similar, so I'm sure there's some similarities, which there are, but there's also a lot of differences as well. So we're going to talk about the differences between RNA and DNA. The first difference is that RNA does contain a pentose sugar. It is in the exact same place that the deoxyribose sugar is placed in the nucleotide. It attaches to a phosphate on one end and attaches to a base on the other. The only difference is that the sugar is just structurally different, and because it is a completely different structure, it has a completely different name. So instead of the sugar deoxyribose for DNA, this sugar's name is ribose, and this is where the R from RNA comes from. Similarly, for the D in DNA comes from deoxyribose, the R in RNA comes from the sugar, which is called ribose. The second difference is that there is no thymine. Thymine does not exist in the structure of RNA. So instead, whenever you want to place a thymine, which would be bonding with an adenine, there is no thymine, so therefore you're going to replace that with a base called uracil. And that would be shorthandedly, you'd just be writing a U, just like you would for thymine. Now remember, there is thymine in DNA. So there is going to be a little bit of a difference, and it may get a little confusing because when we start talking about transcription, we're going to be taking these DNA base pairs and turning them into RNA. So A from DNA will bond with U from RNA, but the T from DNA will still bond with the A in RNA because there is an adenine in RNA as well. So we'll talk about that when we get to transcription, but I just want you to realize that it's only the thymine in the RNA molecule that changes. So A will bond with U, A meaning from A DNA, U RNA, but T will still bond with A. Like I said, we'll talk about that in a little bit. The third difference is that RNA is single-stranded versus DNA being double-stranded. So we've talked about DNA replication. We had to split up the bases and the hydrogen bonds. There is no hydrogen bond that puts bases together for RNA because there is no double strand. The reasoning for that, again, is for transcription. Because RNA is solely used as a messenger, there is no need to make double strands because its sole purpose is just to bond with one side of the DNA and rebond with the side for translation to rebond with anticodons, which will make proteins. So there's no need to have a double strand because we need it to be able to bond with other things in order to make the proteins. And the last difference is that RNA is much shorter in length than DNA. The reason for this is because DNA holds all of the genetic information, but it's not all coded for at the same time, and proteins for every single gene are not coded at the same time. So the reason that RNA is shorter, because it's only coding certain portions of genes, therefore we're not going to be using the entire strand of DNA. We're not going to need the entire genetic code at once. RNA is a much smaller molecule because it's only going to be used for the length that it's going to be coding for. So let's take a look at the structure of RNA. If you see here, we have our sugar, and remember the name of our sugar is called ribose. That's where the R from RNA comes from. The Na from RNA still just means nucleic acid, just as a side note. So here we have our sugar here, and over in the corner we would have our phosphate. So we'd have our phosphate, we have our sugar, and we have our base. So the nucleotide is still set up the exact same way. The only difference is it's a different sugar. So here we have our phosphate. So we have our connected phosphate making up our sugar phosphate backbone. Again, these are all still held together by covalent bonds. Okay, none of that has changed either. And if we look at our nitrogenous bases, this is the set of bases that we have for DNA. Remember, we had adenine bonding with thymine, and we have cytosine bonding with guanine. For RNA, we switch out the T and we make our uracil. So if we look here at the top, we have adenine bonding with uracil and guanine bonding with cytosine still. That doesn't change. Now, just because I want you guys to understand this, if there is a T or a thymine found in a DNA strand, that can still bond with adenine because there is an adenine found in RNA. So A will bond with U, but T can bond with A. Now we know the overall structure of RNA, let's start talking about the different types of RNA. And there are three major types of RNA that play completely different roles in protein synthesis. So the first type of RNA, we have mRNA, which stands for messenger RNA. The next one we have is called ribosomal RNA, short for rRNA. And the third one we have is transfer RNA, and also shortly known as tRNA. So let's take a closer look at these. So the first one we're going to talk about is mRNA. Oops. And mRNA, again, this is messenger RNA. This is going to be our messenger that takes the code directly from DNA, and it's going to be the traveler and go all the way to the ribosome. The ribosome is going to be the major player in protein synthesis because, as we learned about earlier in the year, the job or the function of a ribosome is to make protein. So that's exactly what's going to happen here. The reason why the mRNA is so important is because DNA is stuck inside the nucleus. DNA is not allowed to leave. But in order to get these messages out, in order to get the proteins made, the message has to leave the nucleus and somehow get to the ribosome. So the only way that can happen is mRNA is able to travel. So mRNA will go into the nucleus, DNA will unwind, mRNA will match up the genetic code. Remember, A bonding with U, T bonding with A, C bonding with G, and G bonding with C. And then it will take that whatever code needs to be translated, it's going to take that from the nucleus and travel outside the nucleus and find the nearest ribosome, where that code can be changed or translated from genetic code into an actual protein. So that is the function of mRNA. It's to be the messenger from the nucleus to the ribosome. The next one that we're going to talk about is rRNA, and this is called ribosomal RNA. And the easiest way that we can talk about rRNA is that it makes up the ribosome itself. So we need the ribosome to make protein synthesis. This is the site of where the proteins are made, and rRNA is what makes up the actual ribosome itself. And the third type is tRNA. This is the transfer RNA. This is the major player for translation. So mRNA is really a huge player in transcription and part of translation. It's actually a huge player in both. But tRNA is specifically just for translation. And what tRNA does is it brings what we call an anti-codon. We're going to talk about that in a little bit. But really what it is, it's just a complement to the mRNA code. And along with it, on one side you have the code, and on the other side you have an amino acid. And it's going to carry the amino acids to the ribosome and allow for those proteins to be made on site. Okay, so now we know about the structure of RNA, and now that we've talked about the 3 different types of RNA, we're now ready to talk about the actual production of protein. So this is the start of protein synthesis, and there are 2 phases to protein synthesis, and the first phase is called transcription. So transcription, in the simplest terms I can explain it, would be taking the language of DNA, and by the language I mean A, T, C, and G. Taking those sequence of letters, which, like I said, this essentially is the language, taking the language of DNA and transcribing it or turning it into the language of RNA. And remember, the language of RNA doesn't consist with a thymine, it consists with a uracil. So you're taking the language of DNA, which is A, T, C, and G, whatever sequence those letters are in to code for a particular protein, and you're turning those letters or that language into RNA, which consists of A, U, C, and G. And that's really what transcription is. It occurs inside the nucleus. Remember, the DNA can't leave. So what happens is DNA will unzip very similarly to when it does during replication. So if you see here in this first picture, we have our wound up DNA strand, and here we have helicase coming in, unzipping those hydrogen bonds. Now we have our RNA molecule coming in and adding those complementary base pairs, but remember, A bonding with U, T bonding with A, C bonding with G, and G bonding with C until the message is complete, and that ends transcription. That's all transcription is you're taking that code or that language of DNA and you're turning it into the language of RNA. One more thing to note is that the way that the RNA base pairs are added, it's a very, very similar process to DNA replication, where you do have helicase unwinding or unzipping the hydrogen bonds between those base pairs, But remember, in DNA replication, we said that the name of the enzyme was called DNA polymerase, and that was the builder, and that was the one that was adding all the nucleotides onto each side. It's the exact same concept, except for now, the only difference is the name of the enzyme is called RNA polymerase, and it does the exact same thing, except for RNA polymerase has the capability of adding uracils, where DNA polymerase did not. So during transcription, like I said, you would be taking the language of DNA and turning it into RNA. So here is a sample of what that would look like. So if we have our DNA strand over here, and let's just say the code is ATCGAC. RNA polymerase would add this complementary strand, UAGCUG, because any A from DNA will bond with a U from RNA. Any T in DNA will still bond with an A in RNA because adenine exists in RNA. C will bond with G, and G will bond with C. So the only difference that we haven't talked about from replication is that anytime you would have added a T in the RNA side, you now add a U. But T from DNA can still bond with A. So once transcription is over, we now are going to start to move on to the next phase, which is translation. But there's kind of some rules that we have to follow before we start talking about that. And the way that I'd like to compare the way that the ribosome is going to read, or the tRNA is going to read this message, it's very similar to the way that you would read a sentence. So, you'd always read from left to right. You would never read from right to left. Words are broken up with spaces in between. If there were no spaces between words, it'd be really hard to read a sentence. That's exactly what happens here. So if we take a look at the genetic code, clearly you just see a bunch of letters, right? But these letters, or these sentences, and I'd like to refer to them as sentences, they have words in them. And the way that DNA is able to, or I'm sorry, not DNA, the way that the ribosome is able to read and understand these words is that there has to be breaks in between them. So every single word, I'm going to put that in quotes, all right, every single word is called a codon. So I'd like to refer to as the genetic code as the sentence and the codon as the word. And each triplet, so they're always in pairs of 3. That's what a codon is. It's starting from the very beginning of the sequence. Every triplet set of letters is going to be considered a codon. So this is what we have. Whoops. This is what we have right here. So the first codon would be UCG. Here's our break and our space. Second one would be AUG. Here's our space. Next one is GCA. So in this little sequence right here, we have 1, 2, 3 codons. Or if we're talking about it in terms of our analogy, this whole piece would be the sentence, and there would be 1, 2, 3 words in the sentence. So just as when you know in a sentence that there is a capital letter, that's your signal for, okay, this is the beginning of a sentence. Same goes for RNA. The way that we know that we're at the beginning of a message is there's always a start codon, and that start codon is AUG, no matter what. So ribosome will not start coding and it will not start making proteins until it sees the code AUG. Anything from that point on will be read. There will be an amino acid that's created, and they will continue to link up until you see a stop codon. And there are a couple stop codons. There are 3 of them. So it could either see UAA, UGA, or UAG. You don't need all of them, you just need one of them. So you have to start, and I'm going to go back to this one just so you can see it again. You have to start the message with AUG. There's going to be codons in between that. And then once the ribosome sees either UAA or UGA or UAG, then the message stops, and whatever codons are found after that will not be read, and there won't be amino acids produced after that point. So now that we know that there are codons and we know that those codons code for specific amino acids, well, how are we supposed to know what codes for which specific amino acid? Is there a way to even know? And the answer is yes. And there's actually 2 different charts I'm going to show you here that we're going to be able to use. We're going to be able to almost think of this like you have a Spanish to English dictionary. You're taking the language of RNA and you're translating it into English. And these words here are English words, even though sometimes they don't look like it. These are the names of all the amino acids that can possibly be made. And it, depending on the sequence of these amino acids and how many there are, will depend on which protein is created. Now, just to review a little bit, amino acids, as we recall, are the building blocks of proteins. They are the monomers of proteins. So remember, monomers, the more that you build up, that's going to make your polymer. And the polymer that we're trying to make is proteins. So once you have 2 amino acids strung together, you now have a protein. You have a simple protein, but you have a protein. So this is one of the charts that can be used. Some people like this chart a lot, so I'm going to show both charts. There's I like to call this the square chart, and then there's a circle chart in the next slide. So the way that you would use this, remember you're taking the codons. Codons are in 3 letters. So you always start with the first letter over here on the left, second letter in the middle, and the third letter on the right. So let's just say we were looking for AUG. All right, pretty simple to find that one, right? AUG stands for methionine, and methionine is the name of that amino acid that's coded. And remember, that's also our start codon. So if we were to try to read this and how, if we work backwards, go, what is the code for methionine? Well, you have A as your first letter, so you're going to stay in this box for now. Then you're going to go to U. U is up here, so we're still staying in this box. We're in this row and up here. And then G is down here, so now being more specific, we follow ourselves all the way to the bottom row, and here is methionine. So just to do another one as practice, let's just say I said, okay, in test or quiz, what is the amino acid that codes for CAC? Okay, so CAC, we'll find that. So C is up here, so that means it's going to be one of these over here. We have to find A. A is up here, so now we're in this box. C is here, so we're going to, all right, so here we find this, and our answer for what amino acid is coded for the codon CAC, and the answer would be histidine. So that's how you would use that chart. If we go to the next chart, which this is my favorite, I like to use this one a little bit better. I think a lot of students like this one, but again, it's your preference and they will both give you the exact same answer. This one you work from the inside out. So let's just use methionine as an example, right? So methionine, remember, is AUG. That's our start codon. So we start from the middle. Here's A. Next one, U. Next one, G. And here's methionine. That lands us to that particular one. If we wanted to code for, and I'm just making this up, let's just code for GCC. GCC. We want to know what amino acid will be coded for with GCC. So here we find G, next C, next C, and the answer is alanine. So this one's pretty simple. Like I said, you just start from the inside out, and as long as you stay within the parameters of where it goes, it should easily take you to the amino acid that you're looking for. So now let's start talking about translation. And essentially what we just were practicing by using the codon wheel and the codon chart, that essentially is translation. But let's talk about what's actually happening in the ribosome. So the first step of translation is we need to take that mRNA, the one that we coded from transcription, where we took the language of DNA and we turned that into the language of RNA. Now that mRNA has left the nucleus and it's found itself in the ribosome. So now translation occurs in the ribosome. Transcription occurs in the nucleus. Translation occurs in the ribosome. So now we're in the ribosome. And the mRNA is laid out and ready to be paired. So this is where the tRNA comes in. And if we take a look at this picture, that might be a little bit more helpful. So the mRNA codons are just laying flat. Remember, it's a single-stranded molecule, so therefore there's nothing bonded with it yet. And the job of the tRNA, and tRNA is literally shaped like a T, hence the name, and transfer. The molecule of tRNA has specific codes on it that are complementary to the codon. So this part can get a little tricky. Remember, when we talked about the codon wheel and the codon chart, we were coding the actual codon, the message that came from the mRNA, which is, in this case, this is in pink. It's very important that you don't code for the anticodon. That would make for a completely wrong protein. The only purpose of the anticodon is to simply make bonds with the mRNA molecule so the amino acid can stay in place. That is the only purpose. So we took the language from DNA in the nucleus. Now we are in the ribosome. We have our mRNA laid out here on the bottom. Let's just use these two as an example. AGU is the code. If you go to the codon wheel or the codon chart, you'll see that serine is going to be the amino acid that's linked up to that. And the tRNA is going to find the molecule itself that has a U and a C and an A on it, simply just to bond and keep it in place. And along with that, it's going to bring along serine. And the reason why it does that is because AGU on the codon wheel codes for serine. And then the same thing happens again along with the entire message. So let's just say the next codon is UAC. UAC in the codon wheel will code for tyrosine. So the tRNA molecule will bring in AUG because U bonds with A bonds with U, and C bonds with G. So we have this bonding here. UAC codes for tyrosine. And once all of these codes, we finally hit that stop codon, the serine, the tyrosine, and whatever other amino acids follow after that will link together with what we call a peptide bond, because remember back in the day when we talked about macromolecules, peptide bonds are the bonds that specifically hold together amino acids. They're just covalent bonds that are specifically for proteins, and they will release from the tRNA molecule, and then they will release, and that's going to be your newly formed protein. And from there, that protein will go and it will carry out any function that it needs to carry out. So after you have all of your RNA codes that finally link together, each of these is an amino acid, again linked by peptide bonds. This whole structure here is now your new protein. Here's just another look at translation. So if we look here, all of this down here is our mRNA. These are the open bases that are ready to be bonded. This is the ribosome, or as we like to call it, the rRNA. This is a completely separate ribosome. This is a completely separate ribosome. That's a completely separate ribosome. We're just showing the steps. So technically this is the same ribosome, but we're talking about this in stages. So this would be like stage 1, where we have our first tRNA coming down, bonding with AUG. Remember, AUG is our start codon. And lo and behold, we have our methionine, which is going to be the first amino acid. It's always the first amino acid because it's our start codon. Then we continue, the ribosome continues to move down the line. We now have our second tRNA bringing the next code. Once it bonds, the old tRNA will go away. You slowly start to rack up more and more and more and more amino acids until your protein is complete. You've hit the stop codon, and now your ribosome will float away. The mRNA is going to go back to the nucleus and find a new code. And now here is your protein that you wanted to make, and now it's going to go out into the cytoplasm and it's going to fulfill any function that it needs to fulfill. It's going to carry out the function that it was created for. So just to recap what we've done. We talked about a lot of stuff, and I just want to kind of simplify it a little bit. So remember, transcription happens in the nucleus. This is where the DNA is held and it can't leave. Remember, we talked about replication. That also happens in the nucleus as well. The mRNA, which is also starts off in the nucleus, it's going to bond to the unwound DNA. It's going to code for the message. It's going to take the language of DNA and turn it into RNA. Then it's going to flow out of the nucleus, find a ribosome. Sorry. Then the process of translation will start, where we have our ribosome, which is going to help lay out that molecule of mRNA. Then we have our tRNA, which bonds to the codons, which will carry the amino acid that's going to code for whatever the mRNA code is going to state. Remember, starting with AUG, ending with one of the 3 stop codons. And then your protein is made, and that is protein synthesis.